Hello, everyone. Welcome to today's live broadcast, Statistical Methods for Bulk and Single-Cell RNA Seek Experiments. We are delighted to bring you this educational web seminar presented by LabRoots. I am Judy O'Rourke, and I'll be your moderator for today's event. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker, Christina Kendziorski, PhD. Dr. Kendziorski is a professor of biostatistics and medical informatics at the University of Wisconsin-Madison. I will now turn it over to Dr. Kendziorski for her presentation.
Thank you, Judy, and thanks for the organizers for inviting me to speak. I'm happy to have the opportunity to talk today about statistical methods that my group has worked on for both bulk and single-cell RNA-seq experiments. So this is joint work with a number of my students, both past and present, and the three that were involved the most are listed here. So this work was motivated by a collaboration that I have with Jamie Thompson's lab, and as many of you know, in 1998, the Thompson lab developed a technique to isolate and grow human embryonic stem cells in cell culture, thereby establishing the first human embryonic stem cell line. Over the years, these stem cells have proven remarkably useful for studies of basic biology, especially those concerning development, and they're also a remarkably useful source of cells for regenerative medicine. Now, in spite of their utility, there's much political controversy surrounding human embryonic stem cells, and there's also questions associated with compatibility. And so in part to get around these challenges, in 2007, the Thompson lab published an approach to convert human skin cells back into cells that are almost identical to the human embryonic stem cells, thereby establishing induced pluripotent stem cell lines. Now, if the iPSC lines were not almost identical, but indeed identical to human embryonic stem cells, then they'd be desirable for research for at least most studies because their derivation is less controversial, and also because they introduce the potential to sort of create or develop patient-specific embryonic stem cell lines, thereby attenuating potentially some of the problems associated with immunoincompatibility. But they're not identical, and so a focus in the Thompson lab, as well as other labs that use these lines, is characterizing the differences at all sorts of levels so that we better understand their implications on downstream studies. And so the Thompson lab and other labs look at all sorts of genomic measurements in an effort to characterize those differences, and I've listed some of those measurements here. They look at mutations, methylation patterns, protein. Our group got involved when the questions were associated with identifying differentially expressed genes and also, importantly for today's talk, identifying differentially expressed isoforms between human embryonic stem cells and induced pluripotent stem cell lines. So an experiment that I'll talk about in some detail is given in brief. The outline is given in brief here. We have 4 human embryonic stem cell lines versus 4 induced pluripotent stem cell lines. Messenger RNA was sequenced using the genome analyzer 2. Reads were mapped back to the genome using bowtie, and expression was estimated using RSEM, and this experiment is repeated in triplicate. So the question, again, that we got involved in was, can we identify differentially expressed genes, but also can we identify differentially expressed isoforms in this particular 2-group RNA-seq experiment? So for those of you who might not be familiar, I'll briefly review the background behind an RNA-seq experiment. So in an RNA-seq experiment, messenger RNA is isolated in some tissue of interest. It's reverse transcribed into cDNA, fragmented. The cDNA fragments are then sequenced. Sequenced reads are shown here as colorful lines. They're mapped back to the genome or some reference transcriptome, and it's the data here that's shown on the right, the schematic of that data, that we use to get estimates of expression. Once we have estimates of expression, we go on to do things like identify differentially expressed genes or identify differentially expressed isoforms. Now, excuse me, when we started addressing this problem, there were already a number of tools in place for accomplishing many of these tasks, and this slide is dated, and it's sort of the landscape at the time that we were looking at a few years ago. a number of tools in place for aligning reads back to the genome, for estimating expression, and importantly, for identifying differential expression. Most of them, like DE-seq, EdgeR, BASE-seq, were at the gene level. CUPDIF was available for identifying differential expression at both the gene and transcript or isoform level. So we weren't in the business of making more work for ourselves. We thought that we would apply these methods to our problem of identifying differentially expressed isoforms, and if they worked well for that task, we would have used them. But most of these methods are not appropriate for identifying differential expression at the isoform level, and so I'll spend some time today telling you why. So just about all of these methods, I think all of the ones that I've listed for sure, make common assumptions about the way in which counts are distributed. So they assume that read counts or estimates of expression are distributed as negative binomial with some shape and scale. And here I've denoted that shape and scale as being a mean and variance simply because the shape and scale determine the mean and variance. So you can think about it as counts being distributed as negative binomial with some mean and variance. And the way in which these methods differ primarily is the way in which they estimate the variance. So how is the variance estimated? Most of them rely on some approach that looks at this relationship between sample-specific estimates of the variance, sample-specific estimates of the mean, and they fit some curve that describes this relationship. Okay, so they might use this in different ways, but most of them to estimate variance rely on assessing or quantifying the relationship between the mean and the variance. Okay. The thing about using this is there's only one curve that's used with each method when they're looking at the gene level, and this relationship changes with isoform complexity. So in the isoform case, this curve is a little bit more complicated. So what do I mean by isoform? So that I can define isoform complexity. So here I'm showing you 3 different isoforms coming from 2 different genes. So here I have gene G and gene G prime, and an isoform is just an mRNA transcript that comes from a gene. In the first case, gene G, it's the simplest case. We have one isoform that's unique to that gene. In the second case, gene G prime, we have 2 isoforms. So isoform G1 is unique to its parent gene, and isoform G prime 1 and isoform G prime 2 are not unique to their parent gene. There's 2 isoforms coming from that gene. So NG denotes the number of isoforms coming from the parent gene. NG equals 1 in the first case, we have 1 isoform. NG prime equals 2 in the second case, we have 2 isoforms. And what I'm talking about when I say complexity has to do with how big NG is for a given gene. So if we go back to the slide that I showed you before, the schematic of an RNA-seq experiment, the data that we use to estimate expression, excuse me, is shown just above. I don't think you can see my pointer. It's shown just above this box to get expression estimates, right? So this is the type of data we would use to get estimates of expression. Now, how do you use those reads that are mapping the exons to get estimates of expression? Well, in the NG equals 1 case, it's kind of easy. It's kind of easy. You just use the reads that map to the constituent exons. But in the NG prime equals 2 case, it gets a little bit more complicated because what do you do to the reads that are mapping to exon 2, those blue reads there? You want to divvy them up in a way to the isoforms that's directly proportional to their expression, but that indeed is the quantity that you're trying to estimate. So that is a little bit more complicated. And because that's more complicated, there's increased uncertainty in isoform expression estimates for more complex isoforms. And so let me repeat that. Expression estimates for complex isoforms, those for which NG is greater than 1, have increased variability in their expression estimates because of this problem that I just mentioned. And that increased uncertainty needs to be accommodated if you want improved inference. So going back to the picture that I showed you earlier, here we see variance as a function of the mean, and this is for all isoforms shown in red. Here's the curve if we just look at the simple isoforms, the ones that are unique to their parent gene. Here's more complex isoforms, NG equals 1, and here's NG equals 2, and then shown in green, even more complex isoforms, NG equals 3 or greater. So what you can see is that there's reduced variability in the simple case compared to all the isoforms and increased variability in the others. So if you're using, let's say, the red curve when you should be using the yellow curve, you're using an increased estimate of variability. And so when you think about a test statistic, where we're looking, usually a test statistic for this type of two-group analysis looks like a difference in sample means in the numerator or some function of that, and then an estimate of variance in the denominator. If you're using an estimate of the variance that's too big, then you're underpowered to identify differential expression. Whereas in the other cases, if you're using an estimate of variance that came off that red curve when you should be using one that's in fact larger, you're using an estimate of the variance that's too small. You use an estimate of the variance that's too small, you have increased false discoveries. Okay. So we look to see that. I just showed you that for one data set, and I'm showing this again, like how big of a problem is this really going to be in terms of downstream analysis. We looked at that before we decided that we needed to develop a new method. So here I'm showing you RSEM process data from the Thompson lab. RSEM is one method that's used to get expression estimates. And here, for this region that I have marked on the left-hand side, I'm showing you for all isoforms that have an estimated mean that falls within that region, I'm showing you box plots of the variance for all of the isoforms, and then for the simple isoforms, Ng equals 1, and then the more complex isoforms, Ng equals 2 and Ng equals 3. And you can see that in terms of the median variance, there's quite a bit of difference. We're talking orders of fold change, two-fold change in some cases or more. So let's look at data from another lab. We see the same kind of pattern. We can look at data processed in a different way. So this is not an RSEM effect. Here's RSeq processed data. So RSeq is another method for processing expression data, and we see the same type of pattern. This is MAQC data, so microarray quality consortium data. This is a publicly available data set, not one that we have here from one of my collaborators. Cufflinks processed data. So cufflinks is a method for estimating expression again, and this is another lab's data, and we see the same kind of pattern. So looking at these types of things and then doing simulations where we assessed, okay, when the variance does differ in a way that matches what we see in real data, what is the impact in terms of power and FDR estimated by simulation studies? So we did a bunch of simulation studies and convinced ourselves that in fact this is an issue that needs addressing, and so we developed yet another approach for identifying differential expression for RNA-seq data, but this one was focused on identifying differentially expressed isoforms. It can be used at the gene level, but it was developed for isoform level data, and it was published in Bioinformatics a couple of years ago. So let me give you a bit of background on that approach. Okay, so it's called EV-seq. It's an empirical Bayes approach. And like all of the other approaches, it too assumes that counts are distributed. I'll just underline that here, try to underline it with this funny triangle. Counts are distributed as negative binomial, again, with some shape and scale, and that shape and scale determines the mean and variance. Then we assume that we have a prior that accommodates variability in the true underlying latent level of expression across the experiment. So here's the way the thought process works. We assume that some genes have some latent level of expression. Latent level of expression means we don't know the level of expression. It's the target of our experiment. It's what we wish we knew. It's what we would know if there was no biological and technical variability. It's a true underlying level of expression for that gene. Some genes have low mean. Some genes have truly high mean, and that latent level of expression varies across the experiment, across all the genes. So we use a distribution to govern that variance within the experiment, and that distribution is a beta distribution shown right here. Now, conditional on having a given mean, we assume that counts arise as negative binomial parameterized by that mean. So we can see in this yellow distribution here, there's some variability around some latent level of expression. That variability is due to biological and technical variability. So these are the two components of the model. We have an observational component that describes fluctuations due to biological and technical variability, and we have a prior component that describes fluctuations due to differences in latent levels of expression across the experiment. Now that's the setup for a given condition, but of course we're interested in two conditions. So the top part of the slide is what I just showed you. Again, X, the expression of isoform, given isoform or gene in a given sample S. is distributed as negative binomial, and here I'm showing you the specific parameterization that we use. But I'm interested, of course, on whether or not these latent levels of expression match across conditions. If they do, I say there's equivalent expression and the Q's are the same. Q's determined for a fixed R, the mean level of expression. And if there's differential expression, the Q's are different. So because I don't know which case I'm in, I introduce a two-group mixture model. And I say the data arise out of the following mixture model. There's some probability P0 that there's equivalent expression. There's some probability P1 that there's differential expression. F0 is the marginal predictive distribution under the null of equivalent expression, and F1 is the marginal predictive distribution under the alternative of differential expression. So a couple of things to note about this model. The first is that the prior that we put down depends on isoform complexity group. So this is the main way in which this approach differs from from most of the gene-based approaches, is it specifically accommodates isoform complexity, or the differential uncertainty that we see in different isoform groups. And it's through that prior. Now, if you're applying this approach at the gene level, then ng equals 1 for everything, and that's why this is readily applied at the gene level. Okay, another thing to note is that the form of this marginal predictive distribution under the null is a beta negative binomial. That'll be important to note later on, or maybe just something you want to note for later on. Those are sort of 2 points about the specific model parameterization that we use, but I want to make a few points about mixture modeling in general. The first that we see in this blue box is that I have a two-component mixture model, because there are two options for the ways in which these latent means can sort themselves out across condition. But if I had 3 conditions, let's say, then there are 5 ways in which the means can sort themselves out. They're either all the same, or they're all different, or the first one differs than the other 2, or the middle one differs than the other 2, or the last one differs from the other 2. And so there's actually 5 ways in which latent levels of expression sort themselves out across conditions when you have 3 conditions. And if we do have that case, then we can easily introduce a 5-component mixture model, and we can evaluate in the context of this framework the posterior probability that the gene or isoform falls into any of those groups. So this approach, when lots of approaches say they naturally extend to multiple conditions, but if it's an approach that's maybe t-test based or something like a t-test, when you get into multiple groups, you move into something like ANOVA. And although you can identify in that case genes that have some change, it's more difficult to figure out where the change actually is. Whereas in the mixture model-based approach, you have a specific number, a posterior probability that tells you, quantifies the places that may be different. For this specific isoform or this specific gene, the first condition looks different than the other 2 conditions, and those other 2 conditions look similar. We can actually quantify those possibilities. Second thing to note about this modeling framework is because we make an assumption of conditional independence, the marginal predictive distribution under the alternative is a product of the marginal predictive distributions under the null. So if you don't like the beta negative binomial, or you have a different type of data and the beta negative binomial doesn't hold, but another parametric family will hold, if you can code up the marginal predictive distribution under the null, you can simply provide it as a family in the R code of EBSeq, and all of this other machinery will work for you. EM can be used to estimate parameters, you can check diagnostics, and you can evaluate posterior probabilities of different patterns. So in that way, it's fairly flexible. Finally, I sort of stressed this when I talked about evaluating posterior probabilities of other patterns, but in this context, we can evaluate and quantify the posterior probability of equivalent expression just as easily as we quantify the posterior probability of differential expression. So in some studies, especially the one we're interested in, we don't just want to know with high probability which genes or isoforms are different, but we would also like to quantify genes and isoforms that we know are the same. And in the mixture model framework, both interpretations are equally valid, unlike a frequentist framework. Okay, so to evaluate this, we conducted a simulation study where counts were distributed as negative binomial. So again, we're going to compare to other methods, and this assumption doesn't violate any other methods because everybody assumes that counts are distributed as negative binomial. We have 90% of the isoforms are equivalently expressed, 10% are differentially expressed. We take the form of the variance from edge R, so there might be a slight advantage toward the edge R method because we assume the variance follows the form that they assume in their paper. And parameters and sample sizes were taken to match case study data. So the data looks somewhat realistic, we hope. Excuse me. That's one set of simulations. The second set of simulations is we simulate 80% of the isoforms to be equivalently expressed, 10% to be differentially expressed as in the first set, but the remaining 10% we simulate to have outliers, and the outliers again are sampled from. We take one sample, we multiply it by 4, 6, 8, or 10 again to match what we see in data. So we simulate 200 data sets, 100 with and 100 without outliers. This plot is just put in here to show you for, with the simulated data, we check that many of the features that we see in real data are preserved in the simulation. So here we're looking at the overdispersion parameter, which determines the variance in edge R, excuse me. And we want to see that our simulated overdispersion parameters, or let me say, overdispersion parameters calculated from simulated data are not too different after we add the differential expression component than they are from what we observe in real data, and we can see that that's the case. So the simulation is shown, these are just cumulative distribution functions, the simulation is shown in light blue. Pink is the microarray quality consortium data. It looks a little bit like an outlier because much of that is technical reps, not biological reps, and so our simulation looks reasonable with respect to this diagnostic. And here we have some results. So at the top, we're looking at power and FDR for the isoform level simulation with CupDiff compared with EB-seq. So with CupDiff, they have a sort of a filter where they don't test some genes for various reasons. They may do a pre-processing and determine that this gene isn't, the data at that gene is maybe too variable or it has a potential outlier, and so that gene isn't considered okay. You can override that filter and you can run CupDiff on all the genes, and when you do, you get the results that we show here. Power is 33%, FDR is 0.2%. If you reduce the data set by only considering the genes that CupDiff considers to be okay, you increase power for a reduced set of genes, and FDR is well controlled. So CupDiff seems to have very well controlled FDR, but the power is quite low compared to other methods. EB-seq, in contrast, has reasonable FDR. Our target FDR here was 5%. We're at about 5.2% on average across the simulations with about 72% power. On the right is the same type of comparison, but at the gene level. So we see a number of methods for identifying differentially expressed genes. Again, EB-seq can be used for gene level data, although it was designed for isoforms. We have reasonable power at a pretty well-controlled FDR. And then that was for simulation results, but we also evaluated the approach using publicly available data where there's a gold standard list of genes, and the gold standard is defined by RT-PCR. So this is a list of genes that people think are in fact truly differentially expressed, and we can see that EB-seq works fairly well. And most of these methods work fairly well on this somewhat artificial data set. Okay, so in terms of case studies, I've told you about the first case study in the Thompson lab. I won't go into detail about the results in the Gould lab, except to say that in our paper we show that the results that we see quantitatively in the Thompson lab are reproduced for the Gould lab data. So this is not, the conclusions we draw are not specific to a particular lab or a particular type of data processing pipeline, and we definitely hope they're not specific to just the simulations that we've conducted. We think we've shown that they're not. Okay, so in the human embryonic stem cell and induced pluripotent stem cell experiment, we applied our approach along with two others, DE-seq and EDGE-R, to that two-group experiment, and here's a Venn diagram showing the numbers of genes that were identified in each of those. We're going to take some time comparing the 90 that EB-seq identifies to identifications from EDGE-R. So here's a heat plot of the 144 genes identified by EDGE-R that are not identified by EB-seq, but they are identified by DE-seq as well. And you can see them here. let me see if I can point with this thing. Many of these look okay, but many of them look like they might be outliers in this region here, right? This is in contrast to those that are identified by EB-seq, where many of these look okay, but the ones I'm showing, highlighting in this red line, looks like they might be outliers. Okay. Now, calling a gene differentially expressed because it has an outlier, if it's equivalently expressed but has an outlier, would be a serious false positive that we don't want to make. At the same time, for the data that's in this lower end right here, it looks like there may be differential expression in the presence of an outlier, right? The gene's truly differentially expressed and one set of samples is an outlier. That call would be valid. So we kind of wanted to see if we could delineate between those two possibilities. And I show this part to sort of illustrate how we tried to figure out the differences between a false call of differential expression because of an outlier versus a true call of differential expression in the presence of an outlier. And I also show it because I think it illustrates the power of the mixture model approach when you move up to multiple conditions. So I want to go back to our two-group experiment where we have a two-component mixture model. And again, the number of groups determines the number of components in that mixture model. When you have two groups, you have two possibilities. There's equivalent expression, the means are the same. There's differential expression, the means are different. So here I'm showing you that two-group model in pictures. I have conditions on the left-hand side right here, 2 conditions, 4 replicates within each condition, and I have equivalent expression and differential expression. And so with differential expression, we can see because the color differs between condition 1 and condition 2. That's what I mean by a pictorial representation of differential expression, is the color differs between those 2 conditions. So here's what the 18 component mixture model looks like. And why do I have 18 components? Well, I mentioned that that experiment was repeated in triplicate. So what we're looking at here are 12 replicates from induced pluripotent stem cell lines, 12 replicates from human embryonic stem cell lines. And you can see here's the pattern of equivalent expression. The latent levels of expression are the same across both conditions. Here's differential expression. The induced pluripotent stem cell line, their latent mean differs from the human embryonic stem cell lines. The following 8 patterns represent equivalent expression. All of the conditions have the same expression, shown in yellow, with the exception of 1 replicate that's done in triplicate, 1 cell line that's done in triplicate, and that has differential expression. So these I would consider equivalently expressed with 1 outlier, 1 cell line outlier. And over here, we see there's differential expression. The IPS lines are different than human embryonic stem cell lines, but there's also an outlier present. So these would be valid calls, and over here, valid calls of differential expression in the presence of an outlier. Over here, not valid calls. These are 8 patterns that are equivalently expressed, but they contain an outlier. So we can fit this mixture model to the data, and we can evaluate posterior probabilities of being in any of those patterns to try to figure out if these are false calls due to an outlier or if these calls are okay. When we do that, we see that the genes that I've marked here with a star have a very high posterior probability of being equivalently expressed with an outlier. Now, I should stress that this model is a bit unfair to EdgeR because it's an EB-seq model, so we fit our own model to the data, and so we might be expected to do a little bit better, so then we can just look at the data. So let's just look to see. Here are 9 genes that are called differentially expressed by EdgeR, but they're called equivalently expressed by EB-seq, and the EB-seq 18-component mixture model identifies them as having a very high posterior probability of being in the equivalently expressed with an outlier category. And so you can see for genes like this, I think we would have to agree that this thing looks not differentially expressed. It looks like it's equivalently expressed, but it was probably called because it had an outlier. And although EB-seq didn't have any that came up with high posterior probability of being equivalently expressed with an outlier, we can look at the highest ones just to see what they look like, and here they are. So these don't have super high probability of falling in that pattern, but these are the 9 highest, just to be fair. And these do look a little bit better, I think, in terms of being truly differentially expressed. Okay. And then the final thing I said about the mixture model approach is that we can not only quantify differential expression, that we can just as validly quantify equivalent expression. And that's been of interest to us for a number of reasons. One is we're interested in identifying differentially expressed isoforms that are in equivalently expressed genes. So here's a number of pairs where we find, I'm showing you these are genes with 3 isoforms, and we can see in the 2 conditions, we'll take one here that's very obvious, this one right here. We can see that gene expression looks very similar. This is expression on the y-axis. The gene expression levels look very similar, but the isoform expression levels are different. Okay, so in summary, methods for identifying differential expression at the gene level in an RNA-seq experiment don't work well for isoforms because they don't accommodate differential uncertainty in isoform expression estimation. EBSeq identifies both DE genes and isoforms. It accommodates uncertainty, and it is robust to outliers. I didn't talk too much about that. And it can be used with more than 2 conditions and to quantify equivalent expression. Okay, so we've extended this approach to ordered RNA-seq experiments. And this, too, was motivated by a study in the Thomson lab. So the Thomson lab is interested in this axolotl creature, largely because it has amazing regenerative potential. When you sever one of its limbs, it grows back almost completely. It grows back completely almost throughout the lifetime of the axolotl. Whereas in mouse, we see almost no regenerative potential, even though a number of genes involved in regeneration in axolotl are present in mouse. So we're interested in comparing gene expression patterns in mouse and axolotl at 7 positions along the limb, so sort of proximal to distal along the limb. And we can do this first in mouse, that's the data that I'll show you, and then ask are they the same as in axolotl. So this work was published a couple of months ago in bioinformatics, and it's called EBSeq HMM because we introduce a hidden Markov model to accommodate dependence over time. Okay, so here's what the data looked like. Generally speaking, we have genes over here. You think about a data matrix, and we have multiple conditions, condition 1, condition 2, condition 3, and these are ordered in some way, so it could be over time, or in our case, it's over position along a limb. And we find it useful in this context to introduce 3 types of paths. The constant path, where expression is not changing across conditions. The dynamic path, where expression is constantly changing. So at every transition, we see some change. And then the sporadic path, where there are some transitions of no change and some transitions of change. Those 3 examples are shown here. So to accommodate dependence across the transitions, we introduce a hidden Markov model, and it's quite simple. We have states, and the state space consists of 3 states. Each state specifies some potential change, and we consider the up, down, and no change conditions. So I'm talking about states that describe transitions, so changes in expression across adjacent points. And what we're interested in is how these states sort themselves out across the transitions. So do we see equivalent expression, excuse me, I'm trying to mark this, equivalent, no change, and then maybe up, up, up all the way, or something different? What are those what's that state profile across the transition space? In the dynamic case, for both of these, we introduce an autoregressive component to describe dependence of the expression at time t on previous expression. In the dynamic case, we also introduce a hidden Markov model on the states, whereas in the sporadic case, we don't. Now, because, again, we don't know which condition we're in, we introduce a mixture model over these 2 possibilities, and I haven't shown you the possibility of equivalent expression, but that's also included in there. Now, we use a parametric model that's very similar to EBSeq, but with this hidden Markov component built in. And you can see here the emissions distribution is a beta negative binomial, just as we saw before. And the mean in that beta negative binomial is very similar to what we saw before, except it has this C term here. where C equals C, if S is up, it equals 1 over C if S is down, and it equals 1 if there's equivalent expression. So you could see the way in which this extends EB-seq to accommodate dependence in expression over these ordered conditions. Okay, so simulations were done as before. We have negative binomial counts, 10,000 genes, 5 time points in triplicate. So we simulated 15% to be dynamic genes with some strong signal, 15% with a weak signal, 15% sporadic genes with a strong signal, and 15% with a weak signal, and 40% were simulated to be constant. And then we evaluate EB-seq HMM, and we compare it to DE-seq, which is now DE-seq2 and EDGE-R. And we can see power is pretty good for all of these methods. It's a little bit, it's very comparable when the signal is strong. EB-seq HMM has a slight advantage when the power is weak. So by accommodating dependence over the ordered conditions, we have a slight advantage in identifying genes that are changing, but just changing a little bit, but they're changing consistently. So here are examples of 2 genes identified exclusively by EB-seq HMM. I'm showing you simulated genes here in the upper panel, and then 2 genes identified exclusively by EB-seq HMM in our case study data. Just a second, some things. So I'm getting a question here, thank you, about what's the difference between sporadic and dynamic. So let me go back just a sec. The difference between sporadic and dynamic is that in dynamic, we assume that at every transition, we see some change. And sporadic, we don't assume that. So for sporadic, at a given transition, we could see equivalent expression, and then the next transition, we see up or down. So here, for example, at the first transition, we see up. Let me mark this just a second. This transition is up. This transition is up. This transition is up. This transition is down. This transition is down. There's always some change. Whereas in sporadic, there's no change here. And then there's up. And then there's down, followed by 2 transitions of no change. So sporadic is a mix between a constant path where there's absolutely no change at every transition, and a dynamic path where there is always change at every transition. Thanks for the question. So if you're interested in identifying genes that have some change over time, most of these methods work pretty well. Like I said, we have a slight advantage in power for genes that are changing what we call weakly. Their signal just isn't as strong, but it's consistent. It's consistent across the ordered conditions. But if you're looking to assess where the changes are and what the actual state space is, so for every transition, what is the state? Is it down, up, down, up, down, down, something like that? Then EB-seq has some advantage because, again, it's a bit like the analogy of t-test to ANOVA. With an ANOVA, you can identify this gene is changing across conditions, but you can't quantify too easily. You have to do follow-up tests to quantify where that change is. Is the change between the first 2 conditions, the first and the third, whatever, whereas the mixture model approach quantifies those for you. That's the same for this EB-seq HMM. We quantify the state space across conditions, so you can say with some confidence, I want to see all genes that have a 95% posterior probability of being in the up, 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 down, down, down pattern. And EB-seq HMM provides that, so that's the main advantage of that approach. Okay. Now I want to spend a bit of time talking about single-cell RNA Seek and what we're doing with single-cell RNA Seek. So here is a heat map where we have low expression in green, high expression in red, 0 expression in white for genes along the rows and samples along the columns. And we're looking at the genes that fall between the 60th and 70th percentile of median normalized RSEM expression for these 2 data sets. What are some things that we notice between single cell and bulk? And I should say that we've matched here the library sizes, so we're not looking at really deep sequencing in the bulk experiment and not deep sequencing in the single cell. So these differences are not due to, not entirely due, or they're not due to sequencing depth. I'll say that. There's going to be some effect of sequencing depth, but it's going to be minimal here. It's a good way to say it. So what are the differences that you notice? Well, one is that you see a lot more white in the single cell data, right? We see a lot more zeros. You might notice there's increased variability along the, for a given row, for a given gene along the samples. And I don't think you can see it here, but I'll tell you that there's increased multimodality. Okay, so features of single-cell RNA-seq data that we observe and that other people have observed, there's more zeros for sure, there's increased variability, and for a given gene, there's multimodality, increased multimodality, way more than we see with bulk RNA-seq, and that's above and beyond being just due to the zeros. So I'm not talking about multimodality, that we have a mode at 0 and a mode of non-zero, but even without the zeros, we see increased multimodality. So here's a plot of just log variance for some bulk RNA-seq data sets shown in green, and then some single-cell RNA-seq data sets that are shown in pink, and with and without the zeros. So you can see SC1, SC2, SC3 are single-cell data sets, and then we've removed zeros. And you can see there's increased variance in the single-cell data set. Just a second, getting a question. So the question is, how do you define bulk? And what I mean by bulk RNA-seq is not single-cell RNA-seq. So traditional RNA-seq is what I mean by bulk RNA-seq. Thank you for the question. So in traditional, I'll just back up here. So here we're looking at variance for single-cell data sets and then traditional RNA-seq data sets that are not single-cell data sets. And we can see with the single-cell data, as we might imagine, there's increased variability. With the bulk or the traditional RNA-seq data sets, the signals that we're looking at are an average across 10,000 or more cells, right? And so we'd expect to see decreased variability for that type of data set. Thank you. Now here, I'm stressing the fact or showing the fact that we see increased multimodality with the single cell data. So what we did here is we fit mClust, which is a method for identifying the number of modes in data, and we can see for the traditional RNA-seq experiments, most of the genes have a single mode. This is within a given condition, so we're not looking at differential expression here. Some of them have 2 modes and so on. The sample sizes are comparable, so power should be about comparable to say whether or not there's 2 or more modes. In the single cell data, we can see that we often see 2 modes. Sometimes we see 3 modes and so on. So there's increased multimodality with single cell data. Okay, so there's a number of questions that we and others are working on with single cell data, a number of things that we're excited about with single cell data, questions associated with normalization, with identifying differentially regulated genes. The one I'm going to talk about today is a question associated with identifying oscillatory genes using single cell RNA-seq data. So the challenge for us in this project was to take snapshot expression data, meaning expression data collected at a single point in time, as opposed to a time course experiment where we would look at data collected at multiple times. We have one given experiment, I'll call that a snapshot experiment, and we want to see, can we identify genes that are oscillating when we don't have time course data? So here I'm showing you 4 cells. Again, we're looking at single cell, and I have 2 genes in each cell, and I'm imagining them oscillating. So gene 1 and gene 2 in cell 1, in cell 2, and so on. And these are hypothetical profiles of oscillation. What happens in the single cell RNA-seq experiment is that we collect data at time t, right there. So we don't see these oscillations that I'm showing. I'm just imagining that they're there. Now, if we could get some idea about how long the genes have been oscillating, then we could reconstruct the oscillation, right? So here I order the cells by the amount of time that they've been oscillating. The genes in those cells have been oscillating. So cell 4 has the slowest. It's been oscillating, it has genes that are oscillating the slowest amount of time. Cell 1 is next, then comes cell 3, and then comes cell 2. If I knew those numbers and I ordered by oscillation time here, I could reconstruct the oscillation. One cycle of that oscillation is called the base cycle. Okay. Now, unfortunately, I don't know that, but it's interesting to think about, and it'll come into play in just a minute. What I don't know that. I have data collected at time t here. So how am I going to figure out how long the genes have been oscillating? I can't. If I order by collection order, I don't see anything. And that's not showing up on my screen, but basically you don't see anything if you order by collection order. What I can do is I can figure out, can I order the cells, let me say, in a way that I can reconstruct one cycle of the oscillation, which we refer to as a base cycle. So here I'm showing you 2 base cycles. Because I don't have any time order, I can't figure out, does cell 4 go here, or does it go in a different spot that's identical to it, like near cell 2, right here. I can't figure out those differences, right? But I can get some information on the cell order that allows me to reconstruct one cycle of the oscillation, which we call the base cycle. That order is called the cyclic order. Okay, so how do we do that? Well, first, this is an approach called Osco, and the first thing we do is we screen for candidate oscillators. So independent of order, if you have genes that are oscillating with similar frequencies and you plot one against the other, you will see them showing up as some sort of ellipse. So we fit a model that looks for patterns. We call it the paired sign model, but it basically looks at pairs of genes, and it fits an elliptical function to those pairs, and it pulls out ones where the residuals are small. We call those candidates. So those are oscillatory candidates. Given the candidates, we cluster them into clusters, and the clusters are based on this paired sign model score, so the residuals again, and ones that have small residuals are oscillating with similar frequencies. They may be phase shifted. In fact, they almost always are phase shifted. And then from that, we can recover the order within each cluster, okay, within each group of genes that has similar frequency but is phase shifted. We recover the cyclic order, which is, again, the order of cells that allows us to reconstruct one cycle of the oscillation, which we call base cycle. And we do that by extending an algorithm called the nearest insertion algorithm that was developed for the traveling salesman problem. So with Oscope, we recognize a connection to the traveling salesman problem, excuse me, which is that given a list of cities, you only know the distances between each pair of cities, and I'm showing, pairs of cities below and their distances, and the goal is to find an optimal route that visits each city exactly once and returns to the origin city. And optimal route is going to minimize the overall distance traveled. Here, it's similar to the traveling salesman problem, but we have a gene cluster and we want to find the optimal route through all the cells that returns to the first cell, because again, we are trying to reconstruct an order. I'm just going to move that around, where we start at cell 1 and we move around and we come back to cell 1. So in that way, it's like returning to the origin city. The optimal route is going to minimize what? Well, that's sort of where the extension comes in. It minimizes expression differences between observed and baseline oscillations. Now, the nearest insertion algorithm was an algorithm developed that to address the traveling salesman problem that works pretty well. We extended it to accommodate our specific problem here. So we call that the extended nearest insertion algorithm. Just a sec, I'm getting a question. Okay, so the question is, what is a Bayesian approach? And I think I'm going to get through that, and then I'll answer that question at the end. Okay, so in terms of results, I want to show you what did we do to validate results? Well, we did simulation studies, of course. I won't be showing you the results of that today, but they're in our paper, should you be interested. We also took data where we know the answer from time course experiments. People have done a bunch of time course experiments in a bunch of different settings, as I assume everyone listening knows, and they've identified genes that are oscillating, and then in many cases, they've validated those genes experimentally. So we took a data set, a microarray data set from long ago, where it was a pretty large sample size, and they had validated, the authors had validated a gold standard list of oscillating genes. We ignore the time course order, so we just shuffle the data and we apply Oscope to see what we can recover. And 4 of those genes are shown here. So on the right top panel, we show the known time course order and the actual profiles for each of those 4 genes. And on the left, we show the order recovered by Oscope. You can see these genes are oscillating with similar frequencies. We can recover that. We can also recover the phase shifts pretty well here and here, where it's the highest point in that base cycle that we see for each of these genes. A second validation that we did is we had identified a gene cluster that seemed to be involved in cell cycle, because many of the genes were known to be involved in cell cycle, and so we wanted to see if the order that we recovered using Oscope was consistent with cell cycle. And so the Thompson lab did an experiment where they labeled cells, and they could tell which part of the cell cycle each cell was in, and we're showing here the ENI, extended nearest insertion algorithm, recovered order on the left for 4 genes in that cluster, and those same genes with labeled cells on the right-hand side. So we have cells in the S phase labeled blue, and then G2 and G1, and you can see that the order recovered by Oscope is in fact consistent with cell cycle phases. There are a number of other validations that I won't talk about today. So in summary, and then I'll take the time here at this break before I just close with a few more slides to answer that question about a Bayesian approach. So in summary, as I said in the first part of the talk, EB-seq was designed for inference on identifying differentially expressed isoforms. It also works for identifying differentially expressed genes. Oscope is a statistical pipeline for characterizing oscillatory genes in a snapshot. Single-cell RNA Seek experiment. And we're going to cross this one out for a minute because that's what I'm going to talk about in just a sec. I'm sorry, it's there. Let me just say briefly, a Bayesian method is one in which you introduce a prior component so that you can evaluate posterior probabilities of whatever you're interested in. In our case, it's differential expression. And we assume that the targets of interest, the latent expression levels, are themselves random variables. That's, I guess, a quick way to think about it. Okay, in terms of availability, EB-seq is in Bioconductor, it's in Galaxy, and there's a graphical user interface. The details are in Bioinformatics in 2013, EB-seq HMM, Bioinformatics 2015. What I'll briefly close with is MAGIC. That's also coming out in Bioinformatics in 2015, and Oscope is hopefully coming out soon. So, do I not have? Okay, so that's why that was. Let me just say, I thought I had a few other slides on MAGIC. MAGIC is an approach that we developed for identifying driver mutations in cancer. So a number of methods are available for identifying driver mutations in cancer. The earliest ones are what we call frequency-based approaches, where if a gene is frequently mutated, it has a number of mutations, a very high number of mutations across a patient sample, it is called a driver. The trick with those methods is that you have to figure out how big is big, and so you need to estimate a background mutation rate, and that background mutation rate varies across the genome. So those methods are really focused on identifying good models, developing good models, and estimating accurate background mutation rates so that they can tell that this gene has a number of mutations that's far and above what we would expect given this background mutation rate. That's frequency-based approaches. Then there are another class of approaches that look at the functional impact scores of various mutations, and if a gene is enriched across a patient population for mutations that have very high functional impact, those genes are called drivers. There's a third class about position. So where in the gene have mutations compiled? Are they in important positions in terms of amino acid, in terms of the protein that's produced? And so MAGIC is an approach that unifies those 3 considerations for identifying driver genes in cancer. And it wasn't in my title, but I wanted to talk about it because we're excited about it, and we hope that it can improve the current state of the art in that area. So with that, I will thank you for listening. I'll thank my collaborators again in the Thompson lab, Ron Stewart, Li Fong Chu should be on here, Victor Ruati helped with the earlier work, My former graduate student Ning Lang is first author on the EB-seq work. Keegan Korthauer is first author on MAGIC and is helping with single-cell work now and working on problems in single-cell area, as is Rhonda Bacher. And I am often hiring postdocs. So with that, I'll thank you for your time, and I'm happy to take more questions.
Thank you, Dr. Kendziorski, for that informative presentation. Before we get started on the question and answer session, I'd like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. And I've answered several questions. There appear to be no additional questions from the audience. Give them just a minute. Well, as there appear to be no further questions from the audience, we'll conclude the presentation. Today's webcast will be available for on-demand viewing through November 13th, 2015. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.